Hello dear students, I am Associate Professor of Department of Human Anatomy and Medical Terminology of Azerbaijan Medical University, Anar Abdullayev. Today I will explain you the functional anatomy of liver and pancreas and their development in ontogenesis. Also, I'll talk about the peritoneum, its topography and development. I prepared the plan for easy explanation to you. And the liver or hepar is the largest gland of human organism. It takes place in many processes. In this slide you are looking at the liver from anterior and the posterior projection. It means how is important the knowledge of topography of a organ is very great importance, such as the liver. But yes, we are studying the liver with the digestive system. It's true, because of producing bile, but liver also performs a wide range of metabolic activities required for homeostasis, nutrition, and immune defense. It's important in the removal and breakdown of toxic or potentially toxic materials from the blood, the regulation of blood glucose and lipids, the storage of certain vitamins, iron, and other micronutrients. It takes place in the synthesis of proteins and clotting factors, the metabolism of amino acids, and the bile production. It's involved in a plethora of other biochemical reactions. Since the majority of these processes are exothermic, what does it mean exothermic? It means the exo to outside, thermic means the energy. A substantial part of this thermal energy production of the body, especially at rest, it is provided by the liver. The liver is populated by phagocytic macrophages, components of the mononuclear phagocyte system capable of removing particulates from the bloodstream. It is an important site of hemopoiesis, a formation of blood, but in the fetal period, just in fetus. And you are looking at the liver in this slide. You are looking at the diaphragmatic surface of the liver. The diaphragmatic surface is separated by this ligament, ligamentum falciforme hepatis, which is derivative of peritoneum. It consists of two layers of the peritoneal plates, and they divide the diaphragmatic surface into the larger, bigger right lobe and the smaller left lobe. This is the left lobe. Also look at the passing of the falciform ligament inferiorly. It passes to the ligamentum teres hepatis, this one and this one. Also you are looking at this ligament on the picture. Ligamentum teres hepatis, it is not true serous ligament. It is not the true derivative of the peritoneum. It's former umbilical vein, and it passes through the liver during fetal life. It explains why the liver has the relatively greater size in newborn. In newborn, the relation of the liver to body mass consists of the 5%, because this unique organ, just the liver, receives the arterious blood during fetal life. Other organs, other important organs, including heart, brain, they receive only mixed blood. And the relation to the body weight during adulthood of the liver, just 2-3%, but in newborn 5%. I am repeating. On the visceral surface, you are looking at the visceral surface in this picture. You know that on the visceral surface there are two longitudinal and the one transverse groove, such as the letter H. It means the right longitudinal groove is occupied anteriorly by gallbladder, and the posterior by the vena cava inferior. Left longitudinal groove is occupied by the ligamentum teres hepatis, I explained it, and the ligamentum venosum. Also ligamentum venosum is former vein. Transverse groove is known as the porta hepatis. Porta is translated like the door for entering the proper hepatic artery, and the portal vein and from this door, from this porta, the common hepatic duct, ductus hepaticus communis, leaves the liver and it transmits the bile. The superior surface is the largest and lies immediately below the diaphragm, separated from it by peritoneum, except from a small triangular area where the two layers of the falciform ligament diverge. The majority of the superior surface lies beneath the right dome, but there is a shallow cardiac impression centrally that corresponds to the position of the heart above the central tendon of the diaphragm. The left side of superior surface lies beneath part of the left dome of diaphragm, and the hepar is surrounded by capsula fibrosa with extension that pass into the hepar and transmits neurovascular structures. It's very important in surgical practice. Most of the surface of the hepar is covered by glistening peritoneum viscerale, which is external to the capsula fibrosa. Only the area nuda, you are looking at this picture, which is highly variable in extent, lacks a peritoneal covering. It has a rough appearance because the capsula fibrosa form its surface, and the hepatic veins, the vena hepatica, usually 3, maybe 4 in the number, leave the hepar in the area nuda and thus outside the peritoneal covering. You are looking at the ligaments of the liver. This is the falciform ligament, and at the posterior border it passes to the coronary ligament. On the preparation also you are looking at the ligamentum coronarium, and ligamentum coronarium form at the right and left angles of the liver ligamentum triangulare hepatis dexter, ligamentum triangulare hepatis sinister. You are looking at ligamentum teres. And about the internal structure of the liver, by Quino, liver consists of the 2 lobes, 5 sectors, 8 segments, and also 500,000 lobules. Lobules is the morphofunctional unit of the liver. Because this classification is based on the ramification of 3 important tubular structures of the liver. I mean the hepatic duct, bile duct. I mean also vena porta, portal vein, and the proper hepatic artery. And by this way, each lobe, what is the line which is separated the right and left lobes, this line by Quino passes through the right longitudinal groove from gallbladder and from the vena cava inferior. There are 4 segments at the left lobe and also 4 segments at the right lobe. First segment corresponds to the caudate lobe and is called the caudate segment of the left lobe. Second segment is the posterior segment of left lobe and situated at posterior part of left lobe. Third segment, it's anterior segment of the left lobe. It occupies the anterior part of left lobe. And fourth segment is the quadrate segment of left lobe, and it corresponds to quadrate lobe. The segments of the right lobe very easily can be explained on diaphragmatic surface. You have to look at this picture. The fifth segment is the middle upper anterior segment of right lobe. Sixth segment is the lateral lower anterior segment of right lobe. Seventh segment is the lateral lower posterior segment of right lobe. And again, the eighth segment is the middle upper posterior segment, this one, of the right lobe. These segments form the sectors. There are 5 sectors, and the 3 sectors are belong to the left lobe, 2 sectors are belong to the right lobe. The left posterior sector corresponds to the first segment and is called the unisegmental or monosegmental sectors because the just one segment consists of this sector. Left lateral sector, it's also unisegmental, monosegmental sector. It corresponds to the second segment. But third and the fourth segments of left lobe together form the left paramedian sector. It's by segmentary sector. And the sectors of right lobe are the followings, and the both are the bisegmental. Right paramedian sector, it includes the fifth and eighth segments. Right lateral sector is also bisegmentary, and it includes the sixth and seventh segments. Just beneath the serous membrane of the liver, we can find the Glisonian sheath. Glisonian's capsule of the liver becomes the condensed as Glisonian sheath around the branch of the portal triad structures as they enter the liver parenchyma and subdivided into segmental branches. Thus, each bile duct, hepatic artery, and portal vein is surrounded by single fibrous sheaths, which Kuno called the Wallonian sheath, because after Wallus, an anatomist from the Middle Age who first described the liver capsule. Within each sheath, the portal vein is surrounded by loose areolar connective tissue. It makes the dissection of the portal vein relatively easy. The fibrous tissue around the bile duct and the hepatic artery is tougher, and dissection of these structures is more difficult. This sheath arrangement facilitates surgical control of the right and left vasculobiliary pedicles of the liver, as well as the sectoral and segmental divisions in complex liver resections. Topography of the liver. As I explained before, liver occupies the upper part of abdominal cavity, and mainly this part is called the epigastric region, and this region is subdivided into the right hypochondriac, left hypochondriac, and the proper epigastric region. Anterior superior border of liver locates at the level of the fifth costal cartilage by right midclavicular line. What does it mean, midclavicular line? It's the line which passes through the middle point of right clavicle. Then it continues to the base of the xiphoid process by anterior median line, and at the sixth costal cartilage by the left parasternal line. Posterior superior border begins at the level of the ninth thoracic vertebrae, descends obliquely and locates at the ninth intercostal space by paravertebral line and seventh intercostal space by right midaxillary line. The lower border starts behind at the level of the 11th thoracic vertebrae, runs along the lower border, a lower edge of the right costal arch at the junction of the costal cartilage of the 8th and 9th ribs at the left side. Then it ascends to the point of the junction of 7th and the 8th left ribs. It means that in adulthood, liver locates above than the costal arch, but in the newborn, also in the elderly people, it can locate below than this costal arch. Internal structure of the liver. The morphofunctional unit of the liver is the lobule hepaton. It can be identified by location of the central vein at the center, and at the angles of this lobule we can find the elements of DVA, duct, portal vein, and artery. Within the lobule you can find the two rows, the very interesting arrangement of the hepatocyte inside of the lobules. The two rows of the hepatocyte, between them the capillary which transmits the bile, but the central end of these capillaries are blind. The bile and the blood, they don't mix. At the peripheral part it continues, such as the interlobular bile ducts. And if you are looking at the hepatocyte, the other cells of the liver includes the hepatic stellate cells, also known as the fat storing ito cells, sinusoidal endothelial cells, macrophages of the Kupffer cells, epithelial cells of the biliary tree, cholangiocytes, hepatic stem cells, natural killer lymphocytes, and the connective tissue cells of the capsule and the portal tracts. Kupffer cells are hepatic macrophage derived from the circulating blood monocytes and originate in the bone marrow. They are long-term hepatic residents and lie within the sinusoidal lumen attached to the endothelial surface. Kupffer cells are irregular in shape and have long processes that extend into the sinusoidal lumen. They form a major part of mononuclear phagocyte system, which is responsible for removing cellular and the microbial debris from the circulation and for secreting of cytokines involved in defense. They remove aged and damaged red cells from the hepatic circulation, a function normally shared with the spleen but fulfilled entirely by the liver after splenectomy. You are looking at the gallbladder, at the position of the gallbladder. The boss, the liver and the gallbladder develop from the anterior endoderm of the duodenum. The germ of the liver enters into the anterior mesentery, anterior ventral mesentery, and separates it into two parts. These two parts in future form the ligamentum falciforme hepatis and also the lesser omentum. Gallbladder is the pear-shaped organ which is located on the visceral surface of the liver. You know from your practical classes it has the fundus body and the neck continues to the duct, and if you want the visit card of the duct, and the neck is the spiral like the folds, they form the plica spiralis, and the cystic duct connects with the common hepatic duct, form the common bile duct. Common bile duct, ductus holiodochus, connect with the ductus pancreaticus, and together they open into the mucous membrane of descending part of duodenum. It indicates that both the hepar and the pancreas developed from the duodenum, because we can identify the definition. It is a very interesting moment. The opening of the duct of gland, it indicates the development point of this gland. And also you are looking at some abnormalities of the liver, the riddle-like lobe, when the liver forms the accessory lobe, and the liver also can protrude into the thoracic cavity, form the anomalous lobe. The abnormalities of the connection between the cystic duct and the common hepatic duct. For example, in the B variant, you are looking at the fibrous connection. On the C variant, there is no cystic duct and the common hepatic duct continues like the common bile duct. Junction with the right hepatic duct, very interesting moment, and also maybe you are like the future surgeons will observe these variations in your patients. Cystic duct in front or behind the common bile duct. Also variation of the cystic duct, the connection with the liver. You are looking at the very long cystic duct and the common hepatic duct and very short common bile duct. Fibrous junction between them. High union with common hepatic duct, very short the common hepatic duct and the cystic duct, and very long common bile duct. Cystic duct in this case is absent or very short. Also anterior spiral joining of common hepatic duct on the left side, and the posterior spiral joining common hepatic duct on the left side also. Anatomy of pancreas, it's a very interesting organ with both secretion, exocrine and endocrine secretion. It locates on the posterior wall of the abdominal cavity, being the extraperitoneal organ. It has the head, caput, neck, body and tail, and the head of pancreas is totally surrounded by the parts of the duodenum. Its weight just 70-80 grams, and you are looking at the inner structure of the pancreas. Its duct originates at the tail ratio and continues to the body and enters into the descending part of duodenum. Before it is connected with the common bile duct, and at the junction they are surrounded by the muscle musculus sphincter ampullae hepatopancreatice. It means that this muscle regulates the entering of the bile and the pancreatic juice into the duodenal lumen. The variation in the ductal anatomy of the pancreas very interesting. As I told you, pancreas, like the liver, develop from the endoderm of the duodenum, but from the two germs, anterior and the posterior germs. Then they connect together, form the the whole pancreas. You are looking at this variation. The main pancreatic duct is formed by fusion of the dorsal and ventral bud ducts. Bud mean the germs, and communicates with the accessory pancreatic duct. It's the variant of the A. B also normal. There is no communication between the main duct and the normally site accessory duct. Pancreas division. The majority of the pancreas drains via a dominant dorsal duct, yes, and that enters into the duodenum but at the minor papilla. A short ventral duct is seen draining along with the bile duct into the major duodenal papilla. Absence at the variant of the D, absence of the accessory duct. The exocrine tissue you are looking in this micro preparation at the acini of the pancreas, and also islets, very interesting islets of the Langerhans. They are endocrine organs, they are endocrine part of the pancreas. They are surrounded by the capillary network. During our lecture about the endocrine system, I will note that the one of the main features of the endocrine system, it's the very great, very rich capillary network around them. And about the cell types of endocrine system: alpha cells, glucagon, beta cells, they produce the insulin, and the somatostatin, gastrin are produced by delta cells, and the pancreatic polypeptide they produced by the F cells. What is the important in this moment? The glucagon and insulin, they are important in the carbohydrate exchange of the body, because by the insulin the glucose with the excess glucose storage in the liver, but again the opposite process when the glycogen, when the organism needs the more glucose under the process of glucogenolysis, under the force of the glucagon, it enters into the blood, and this process delta cells and the F cells are less common and secrete the somatostatin and pancreatic polypeptide respectively. Epsilon cells secrete the ghrelin. Ghrelin, very important, very interesting hormone. It has the very high level in organism in the blood when the human wants to eat. After the process of eating, the level of the ghrelin becomes the lower. And what about the peritoneum? Peritoneum is the largest serous membrane of the human body. You know, it has the two layers, the parietal and the visceral. Before explanation of the peritoneum, I want to begin from the development. From last semester, from the material of the last semester, you know the anterior part of mesoderm doesn't undergo the process of segmentation, and it formed the splanchnotum. Splanchnotum has the two plates, splanchnoplevra and somatoplevra. So splanchnoplevra covers the organs from outside, and the somatoplevra the body cavity from inside. By development of diaphragm and also by dividing of the anterior plate of the mesoderm into the somatoplevra and the splanchnoplevra, the primary body cavity of embryo divides into the four cavities. There are the two pleural cavities, the pericardial cavity, and the largest one, it's the peritoneal cavity. Peritoneum has the sex differences because it is closed from the lower side in males and it's open in females by the uterine tubes, uterus, and the vagina. It can connect with the outside, with the external environment. And the peritoneum has some derivatives such as the serous, ligaments, omentum, lesser omentum, and the greater omentum, and also mesentery, you know from your practical classes, such as the colon transversum, colon sigmoideum, sigmoid colon, transverse colon, jejunum, ileum. They have the mesenteries, and between the two plates of the these mesenteries, the blood vessels and nerve reach the corresponding organs, which is important in the nutrition of this organ. Peritoneum covers the abdominal cavity from the inside and it's very rich with the lymph capillaries, but the visceral peritoneum is very rich with the blood capillaries. It means that by the visceral peritoneum some liquid is produced, but the parietal peritoneum is resorpted. It indicates that in peritoneal cavity there is the some amount of the liquid, peritoneal liquid. It's important for the motoric activity, for motility of the intestines, for example. And also peritoneum form the special folds when it covers the structures of, for example, anterior abdominal wall, the beneath the umbilical ring. It forms the plica umbilicalis mediana, plica umbilicalis medialis to urachus, to umbilical artery correspondingly. And at the upper part, upper storage of peritoneal cavities, there are the hepatic bursa. Bursa means the sac. It includes the right lobe of the hepar and the gallbladder. Again, the pregastric bursa, it includes the left lobe of the liver and the spleen. And the omental bursa. Omental bursa, it is located behind than the lesser omentum and the stomach. Posterior wall, it's formed by the pancreas. What's important, for example, during the operation, you can palpate by this green arrow. If you pass, it is the foramen epiploicum for passing of the finger or the other structures from this opening from the hepatic bursa to the omental bursa is very important. And this is our references of this lecture. I want to thank you for your attention, and I want to say if you will have any questions, you can contact with me. Thank you for attention.